So the presentation is around blind sample size estimation in early phase studies, actually in the context of biosimilars. And so if we are in the concept of biosimilars, forget about the phase 1 being called early, because in principle it's a key pivotal study in your development plan. Usual disclaimer, that's not important. The most important is, actually, I'm a spokesperson here. The work has been done or the case is really a case study that we have faced in our developments, and different people from Merck, the stat group, as well as from Cytel has really worked together on challenging questions received from the health authorities. And essentially what is going to be shared here in between the lines is some of the challenge that we have faced in some cases, not in others, which at the end shows that the way statisticians might look into what is being proposed is sometimes biased because we have some areas of focus or some concern that others do not have. So in terms of the outline, so it's really background and purpose, what we face as statistical challenges in blind sample size estimation. I'll go through a case study and then go through some learnings and conclusion. So background around biosimilars development for those who are not familiar. Essentially, you have a candidate biosimilar that you want to evaluate compared to an original medicine. And essentially, your development is in 2 steps. You have your phase 1, mainly in terms of PK/PD, that is to demonstrate similarity in the PK/PD profile of the biosimilar. And then if you manage to demonstrate similarity, you move to a phase 3 where it's more about efficacy and further safety evaluation. So the part here, when in the example that we have faced, is actually example in phase 1, not in phase 3, but the same kind of challenge might come if you go there. One of the typical design that we have in those phase 1, not much different than in any other development. It might be either a parallel design or a crossover design. Again, as we will see, we have had both cases with parallel design and with crossover design. So when you test the comparability between your biosimilar and the control medicine, it is a classical bioequivalence or equivalence testing that you can play with a 2 one-sided tests. It is the case with a biosimilar. Go back to my history when you evaluate vaccines and you add on some antigen and you want to evaluate the equivalence of the combination. It is exactly the same kind of problems that you have there. And in general, there is an acceptance with some difference between FDA and EMA, but that the similarity is demonstrated if with a type 1 error of 10%, you have your 90% confidence interval, which is in the margin of 0.8 and 1.25 for a ratio scale. Typical PK parameters that we have to consider in those phase 1 studies to demonstrate the similarity is the AUC, the area under the curve, as well as the Cmax of the profile. Now, I was saying we might have both crossover and parallel design. In general, you come to parallel design when you have the long half-lives of the product, or you go to crossover otherwise. Your sample size is going to be driven by the treatment effect ratio, And it's often assumed as 1. And then if you want to be a bit more conservative to ensure some level of success, you're going to actually take a more negative or a more perceived negative expectation or expect value of your ratio and take a 0.95. And obviously, the sample size is dependent on the level of variation, the level of variability with the coefficient of variation when you look at the ratio. So the challenge. I mean, this slide, I can use it for any time we look at using some flexible design. But actually, as a stat, it's really an interesting perspective. The left-hand side here is actually a project management concept where you have 3 dimensions. You have the quality, you have the cost, and you have the time, with always a challenge that you all face. You can handle 2 of the dimensions, but not a third one. So it means that if you want to have sound results, okay, either you have to accept that it takes time, while you have low cost, or you want to speed, and that increases the cost. And then you can play with a different dimension, but it's an interesting tool, notably when you have to discuss with clinicians around more flexible design, that the first perception and the first barrier is to say, oh no, I understand it's give more room, but actually I'm going to have to interact with the authorities, and that will slow the start of the process. And the challenge being there that often people think at when we start as opposed to when we end. Now, the key point, which is to remind, is actually a study that fails is not a study where you don't reach the primary endpoint, but it's actually a study that does not allow you to take a sound decision to move to the next step. And that's an important element because as well, when you try to find some flexibility in the way you drive your design, it's actually to ensure that you have an answer to your question at the end, as opposed to have light information that does not allow you to take some decision to move forward. So typically in the type of biosimilar development in those phase 1 with PK parameter, what we face is that there is a high variability, and as well, the level of knowledge of the variability is limited. And we know how in phase 1, moving from one syndrome to another is actually, you get some fluctuation in the variability estimate. And obviously that induces uncertainties about what is the optimal sample size and how you actually have to have the optimal design to reach your or address your study objective in a meaningful way. So you increase the risk of failure. And that's where the blind sample size re-estimation come into the game. For those being familiar or not familiar, you can either go for an unblinded sample size re-estimation or a blinded sample size re-estimation. In general, when we look at the perspective of the health authorities, what they definitely prefer is that you go for a blinded sample size re-estimation to not influence your decision or the change in the design based on the actual treatment differences. So it consists at recalculating the sample size, need to achieve a predefined power level. So there is a certain control in the process on the basis of what is the level of the variability being observed on blind data after accumulation of a certain number of data. Usually, so it takes place with a subset that is initially planned again. And in the literature, you will find out that it's also named as the internal pilot study to the trial. So when it's the variability that is the level of uncertainty at the design stage to maintain the power of the study. So the point here is, again, when we don't know and that there is a certain variability or a certain unknown about the nuisance parameters, how we can actually reassess to reevaluate the sample size. So it leads to a re-estimation of the sample size to that predefined maximum number of subjects. And actually, there are two approaches that can be applied, which is either the unrestricted design or the restricted design. They both actually have the difference in the way the final sample size can result. So in the restricted design, you actually, you always are at least as large as the initial planned sample size, as opposed to the unrestricted, where it will be the maximum of the interim or interim assessment sample size, and the it might be less than the initial planned sample size, while the restricted will not. And as such, the restricted design is generally preferred because it bounds better the change you might do and not come with an approach where suddenly, based on the variability, you will have much less data being accumulated. So on a regulatory standpoint, it is clearly preferred. So if we are back to the biosimilar and the equivalence testing, you come to your null and alternative hypothesis, the classical one that comes to the two non-inferiority hypothesis. And you can, from there, reject the null hypothesis based on your T distribution if you assume the initial normal distribution and difference between the two treatments. Now, the variance is the pool variance estimate, and as we will see, this is part of the challenge when we come to sample size estimation in a blind way, because the sample size, the pooled estimate of the variance is actually based on a pooled average, okay, as opposed to the two separate mean for the two treatments. So from the non-inferiority testing, you can then derive the way to estimate the total required sample size for the rejecting the null hypothesis. And you can either use an exact or an approximate method. Here in the approach, we looked at the approximate, which is the one published in the literature. So the issue faced is that when we come to non-inferiority or bioequivalence, we face an issue on the way to control the type 1 error. So in superiority trial, there is no inflation of the type 1 error. And this is linked on the way the null hypothesis and the alternative hypothesis are being defined, which is somehow the opposite towards the equality of the two treatment arm. So in superiority trials, we don't face inflation of the type 1 error. But when it comes to non-inferiority or equivalence, the nominal significance level may be exceeded. And that becomes the regulatory challenge. So Fried and Kieser in 2003 came with a proposal in terms of the methods. And they initially came with the example for parallel design. And they come with analytical calculation of the type 1 error with the assumption of the normal distribution. And their key idea is the way they managed to go there is actually to decompose the test statistics that we saw before into the different component, and from there derive the joint density distribution. So the way they did, they had Z1 being the standard normal distribution, the standardized normal distribution for the data accumulated up to the sample size re-estimation point, and then Z2 being the standardized Z statistics based on the incremental data after the sample size re-estimation. And they derived the two variants. So you have actually V1, you have V2 that is there, but then with some correction to account for the accumulation of data. And they show that under the null hypothesis, you have those different distribution that can be derived, while actually for V2 star, which is linked to the bias, relate to the pooled estimation of the variance. You can approximate to a chi-square, but it's an approximation. So from there, you have your joint density distribution, and you can as well decompose the variance estimator, okay, using Z1, V1. And so the sample size recalculation can be based on Z1 and V1 and the true required sample size. And so from there, you can come to the actual type 1 error rate. And so they come to an actual alpha, which is the actual type 1 error you might choose to account for the inflation of the type 1 error. Okay? Quite technical, we go with an example. After the initial paper, from Frank and Kisher, actually you had two other important papers. So the Golkowski paper with Frieden and Kisser as well, they extend to the case of a crossover design, where you add some complexity in the estimation of the variance. And then Shen published as well a paper using exact formula for the sample size re-estimation at interim. And so what we did, we investigate both approach using numerical calculation to see in the crossover design case we had to which extent using one method or the other was important on one hand, and on the other hand, how we could come up with a type 1 error that was containing the inflation and that would be acceptable on a regulatory standpoint. So in the example, we had a crossover design with, again, primary endpoint being the AUC, the Cmax. We had as best estimate coefficient of variation of 25%, and we came out to 80 evaluable subject per arm to needed, assuming a 5% between, so the 0.95 ratio. And we came up to that sample size with an adjusted alpha to 4.6 as opposed to the 5, okay? The one-sided 5. To illustrate, not in terms of sample size, but in terms of power, if the true viability is increasing, you see how your power is decreasing. And again, it sounds crazy outside biosimilar to power a phase 1 at 90%. Just as a reminder, your biosimilar phase 1 is almost like a pivotal study, so you target to power at a higher level. So we evaluate different scenarios for the sample size estimation here after 50, 104, 150 subjects. And we came to a maximum number of subjects to be set at 214, so with a maximum coefficient of variation. And the table here shows with the example of a sample size estimation of 104 at 104 subjects using the Golgoski approach. You see here that is the chosen type 1 error, and this is the actual type 1 error. So that's the nominal, and that's the actual type 1 error. And so you see if you have 4.6%, you go at some point, depending on your coefficient of variation, you go to 4.7. If you take a 4.7%, you go to 4.8, 4.84. If you go to 4.8, you are close to 5%, and then if you go to 5%, depending on your coefficient of variation, you actually go above 5%. Now, when you see those numbers, you just, if you step back and you say, okay, I mean, it's a joke, okay, in a way, because it's so close. In the approach we had for this trial to the FDA, there was some debate about, okay, where we put the bar here. And we chose to take a conservative approach, and we proposed a 4.6. Okay? And then I'll explain what happens. So if now we represent the table more where we see the different coefficient of variation and the actual alpha, okay, for different nominal alpha. So you see how your actual alpha is evolving depending on the coefficient of variation, okay? And somehow when the coefficient of variation is small, or that, I mean, you can translate the other way, that the equivalence range is wider, you never need to increase the sample size, almost. Because the design behaves like a fixed sample size, because your maximum sample size will be driven by it. If you have a large coefficient of variation, or it's almost like the equivalence range is narrow, you always increase the sample size to the max. And so again, it almost becomes behaving like a fixed sample size. So that is another way to explain how in the tail, somehow, you reach your nominal alpha. Then we look at comparing the approximate methods approach, approximate distribution, approximate sample size estimation, and the approximate statistics with the normal distribution assumption with the exact one, so the paper from Chen. And there again, if you compare the two tables, I should have represented in a better way because there is all those numbers, but the figures are very close to each other again. While on the other hand, when you go into the exact density, you come with much more challenging and more intensive computation and simulation being needed. So some of the learnings and conclusion, when we started, we didn't pay too much attention to the fact that for non-inferiority or equivalence, you face the challenge of the inflation of the type 1 error. The magnitude of the inflation obviously depends on the required sample size, the sample size that you, for the internal pilot study, and the equivalence or non-inferiority margin. So we saw the example, with notably with the graph. An adjusted alpha level is to be used, and especially if the study is a pivotal one. If we look at the methods and the initial methods, you have two approximations that are possible when assessing the type 1 error. Rate for the sample size estimation. On one hand, the test statistics, so the t and the normal, and the sample size calculation. And what we observe is that the difference in terms of sample size calculation and the type 1 error rate when using the approximate compared to the exact methods were very small. Now, it's clear that when you come into area where it's more sensitive, and if you go down in terms of the sample size, then you might come and start to see more difference between the exact and the approximate method. One of the part of the challenge, or part of how we are challenged as well, is actually the numerical stability of the estimation, because all the integrations are done numerically, and somehow the health authorities want to have more control around the stability of the estimation. That ends my presentation, and happy to take some questions or to share more.